This is the final presentation on the topic of hormonal communication, and it is diabetes, and this of course follows on from the previous presentation on control of blood sugar. When we look at our specification, we can see that you're supposed to study the type 1 and type 2 diabetes mellitus, including the causes and the treatments for each, and also we'll be looking at the ways in which insulin is produced, and we'll be talking about how we use genetically modified bacteria for that, and that will also link into the biotechnology course that we'll do later in year 13. So our introduction then for diabetes, most of this stuff will be very familiar to you, but we'll just quickly run through it. The body has to keep the blood glucose concentration constant. That's because the cells need a constant supply of glucose, and particularly the brain. The brain is very sensitive to low glucose, so actually the system is set up to try and maintain a constant blood glucose concentration of around about 90 milligrams per mil, 100 mil, sorry. Insulin and glucagon are the hormones which coordinate this process, managing to increase or decrease the glucose as it's required. In around 300 million people, the system doesn't work properly, and if that's the case, they are said to have diabetes mellitus. Type 1 is the first type of diabetes that we need to cover. This is where somebody is unable to produce insulin. The beta cells are not producing insulin, so they have become damaged in some way and they are no longer producing the insulin that you need. So you have a meal and your blood sugar is increasing because you're absorbing the glucose from the food that you've digested. Blood sugar is increasing. However, though that glucose is circulating past the pancreas is unable to detect that rising glucose, and so no insulin is being released in response to that. So the glucose levels continuing to rise in the blood. This has quite a lot of impacts upon the system. Firstly, because we've got more glucose in the blood pressure is going to rise because glucose lowers the osmotic potential of the blood and draws water in and hangs on to it. The fact that we've got no insulin means that the glucose is now no longer being stored as glycogen, so we are not keeping any of that glucose in reserve. So we're getting less storage of the glucose and we're using it much less efficiently. A lot of that glucose is circulating in the blood and it will be excreted in the urine because the kidneys will filter it out because it can't reclaim all of it in sufficient time. So all of this means that the levels of glucose in our blood are going to stay high. If we run through some of the symptoms that you would experience if you suffered from type 1 diabetes. Now this is to do with our high glucose concentration. So the first thing you might find is that you're feeling tired. Now of course this is a very general kind of symptom, and often people don't recognise this one straight away. But feeling tired is because we're actually low in energy, and that's because we don't have any energy supplies or any reserves in place because glycogen isn't being stored. And obviously we're not storing any of our glucose as fat or anything like that either. Because we've got so much glucose in our blood, when the blood passes through the kidneys, the glucose is hanging onto a lot of the water, and not all of that glucose is going to be reabsorbed in the kidneys, so it remains in the filtrate, and that then leads to larger quantities of water in the filtrate, which means that the body excretes the glucose but also takes with it quite a lot of the water. So somebody who is diabetic will need to go to the toilet quite frequently. Weight loss is a very common symptom of type 1. So because of the way that this is working, because the body is no longer producing insulin, its glucose is not being stored as glycogen, and it's not being stored as fats either. So the body is not making any stores of these things, but also it's actually has to use up its fat stores in order to keep the blood sugar levels high. So a person will start to drop weight. Wounds that won't heal. Somebody that is diabetic may find that they get cuts and so on, and they don't get better for a long time. And this is basically because the blood, as it circulates, is carrying so much sugar, and this provides a really good raw material for the bacteria to thrive and grow. So bacteria and fungi grow very well in blood like that. This can be a huge problem for people in terms of their feet, because circulation tends to be a little bit less good in your feet. So people with diabetes often end up with damage to their feet, and also wounds that don't get better, and it can lead to really serious infections like gangrene, and people quite often end up having toes and so on amputated because of it. Always feeling hungry. Obviously you're losing weight, glucose that you eat is going out, it's going down the toilet, so you're going to have a need to keep on eating, so you're going to be eating a lot. The increase in glucose in your blood causes a rise in the blood pressure, because it causes the blood to increase in volume, because we've got lower water potential. So that can lead to high blood pressure, and that can affect the blood vessels in your eyes and can create certain types of vision problems. The high blood pressure and the effect of glucose can also cause damage to your nerves. So nerve damage can be recognised as numbness or tingling in the hands and feet, and obviously a very serious condition. Always being thirsty. Of course, you're producing lots of sugary urine, and that then means that you're losing large amounts of urine, so the body is going to need to replace that by feeling thirsty all the time. There is some evidence to suggest that type 1 diabetes is in fact an autoimmune disease. We mentioned this when we did the health topic, and this is where the immune system is actually attacking the beta cells and destroying them, and it's usually something that develops in sort of mid-childhood, sort of perhaps about 9-10, but it can happen even earlier than that. And so this, though having said that, it can actually happen pretty much at any point in your life, but it is much more common to develop it as a child. In the case of type 2 diabetes, people cannot effectively use the insulin to control their blood sugar levels. This might be because the beta cells are not producing enough insulin. Alternatively, it's because the body isn't responding to the insulin that's being produced. Now, in fact, what happens is, for someone who is type 2 diabetic, glucose levels rise in the blood because the person's eaten a meal and they absorb glucose into the blood, and the glucose levels go up. And not enough insulin is being produced, and so the glucose levels rise. The other possibility is that the body is producing insulin, but it's not responding to it. So either way, the insulin isn't having the effect it should have, either because the body isn't responding or it's not producing enough, so glucose levels continue to rise. Now, in fact, when the body is producing insulin, but the body isn't responding, what it does is it actually causes the pancreas to start to produce even more insulin. So you can actually detect type 2 diabetes with somebody by actually measuring their insulin levels. If they've got very, very high insulin levels in their blood, it suggests that they are developing type 2 diabetes, because the pancreas is starting to produce huge quantities of insulin to try and get the organs to respond. And that lack of response just encourages the pancreas to make even more. And eventually that can lead to the pancreas not being able to make any insulin at all, and it's kind of just packing up, and then eventually they're not producing enough insulin. Type 2 diabetes is most usually caused by people because they have too much body weight. So being obese is making much more likely that you will develop type 2 diabetes. Being inactive as well, which obviously will be linked to obesity, and overeating of carbs, so eating far too many carbohydrates and sugary foods. All of those things, of course, are linked, and all of those are likely to lead to a BMI that is obese. And if that is the case, then you're much more likely to develop type 2 diabetes. And of course, this has been linked with a range of other conditions. Being overweight and having type 2 diabetes can also lead to a whole range of other health issues. And if we look here, these are some of the symptoms that you would experience. You tend to develop these conditions very slowly, so a lot of people will not necessarily recognise these things because they're happening gradually. And it's becoming a problem which is much more common with age, but it's much more frequent in childhood. So you can see all the same issues that we mentioned with type 1 diabetes, but because the onset is much more slow, people tend not to notice it quite so easily. And it is thought that body cells are not responding because the glycoprotein receptors, which would be on the surface of cells, are not working properly. Now in this diagram what we can see is the glucose and the insulin circulating in the blood. The insulin is, on the second diagram, the insulin is attaching itself to the receptor there, and that's opening up the glucose channels so the glucose is able to get in. So somewhere along the line the cells are not responding because the insulin isn't able to bind to the insulin receptor. And there has been some suggestions that some of the reasons for that is that there's so much fat coating the internal organs particularly that it makes it very difficult for the insulin to reach its target, because it's really just the target isn't accessible. It's too much fat surrounding those internal organs. Interestingly enough, there is some work to show that people who are obese are much more likely to have this problem, but there are also people who are known as slim on the outside and fat on the inside, and those are people who don't necessarily look terribly overweight, but the way that their fat in their body is distributed means that the fat is coating the internal organs and making it much more likely that they will develop type 2 diabetes. So it isn't always because people are incredibly overweight that they develop type 2 diabetes. Sometimes it's just the way in which the body metabolizes and stores the fat. So you may not necessarily look hugely overweight. So obviously what we need to do is treat these conditions, and treating type 1 is a relatively straightforward process. It's not curable, but we can control it with regular injections. So treating type 1 diabetes involves regularly testing the blood sugar levels, and there are a number of ways in which that can be done. The first one is by using a machine which gives a little bit of, pricks your finger and you get a little bit of blood, and that analyses the level of glucose in that blood. And then from that information, when you know how much glucose you've got in your blood, you know how much insulin you need to take. On the right there, you've got a slightly different type of system. It's called a flash system. And this is a much more modern system where you have a monitor which is placed underneath the skin, and that monitor is continually measuring your blood glucose, but you can only find out what the blood glucose concentration is if you scan it, and you can use, quite a lot of them use your phone to do that. There is actually another type of monitor which is a continuous monitor, and again it's worn by a sensor, but it's connected up to a different system which is continuously recording your blood sugars, and that can give you quite good information as to how your body is responding over a longer period of time. You can work out where your blood sugars go up and down throughout a 24-hour period, so you can start to get into some kind of routine. Once you've worked out how much insulin you need to take, then that insulin will get into the system, and what it will do is it will increase the amount of glucose that's absorbed by the cells, so that causes your glucose levels to fall. So it stimulates glycogenesis, and your glucose will fall to a more reasonable level. Now of course this isn't being controlled by the two hormones monitoring the blood, so it will just keep falling as long as the insulin is there and working. And don't forget, we mentioned that insulin is once it's done its job, it's broken down, so it isn't going to keep on causing that fall in blood sugar, but the more insulin you take, the greater the drop in blood sugar will be. So if you take too much insulin, you could lead to very low blood sugar, and that's called hypoglycemic. And that can be very serious. A real fall in your blood sugar can result in somebody becoming unconscious, and then they could even die. People are trained to recognise when their blood sugars have gone too low, and they will be encouraged to carry around with them some sort of sugar form, so maybe some sweets or a sugary drink, something like that, which they could take very quickly to just push their blood sugars back up a little bit. That's why it's so important to keep on monitoring your blood sugar. On the other hand, if somebody injects too little insulin, you could end up being hyperglycemic, so having too much sugar in your blood. Now, of course, before you're diagnosed with diabetes, you are hyperglycemic, so you're going to be suffering a lot of things, all the symptoms that we mentioned before. But if that goes on for too long, then this can result in very serious damage, which can make a person go unconscious, and they could die from that as well. Treating type 2 diabetes is a slightly different issue. The first thing is for a person to try and regulate the quantity of carbs that they are eating. The secondly is to increase the amount of exercise. So obviously most of the time it's going to be more exercise. And thirdly, you need to lose some weight. So because type 2 diabetes is very much linked to diet and levels of exercise and weight gain, you need to try and reverse those things as much as possible. There has been a lot of research in recent years that shows that people can actually put diabetes into reverse if they lower their carb intake and actually lose a lot of weight quickly. If this doesn't work, then people can be given some drugs which may help. So some of them might be stimulating the insulin production, and some of them might cause the rate at which glucose is absorbed to be slowed down, so that it actually reduces the amount of glucose that enters into the blood. And thirdly, you can actually start to have insulin injections. You have this table to help you summarize some of the information that we've given you on these two types of diabetes. So I suggest that you summarize it as you can in here. Now we're going to just go over how insulin is produced. So this is actually work about genetic engineering, which we're going to be covering again later in the year. So you won't have any notes on it on your worksheet, but I felt it's quite good to try and link it up at this point. So obtaining insulin for treatments then. Originally, insulin was extracted from pigs because pigs are very similar to humans in many ways, but that did have its drawbacks. Firstly, it's quite difficult to get the insulin out of the pancreas. It also was quite expensive, and people quite often suffered allergic reactions, and that's because although it's very similar to human insulin, it's not exactly the same. We now produce most of that insulin using genetically modified bacteria. So we're just going to run through the process of doing that. This is something most of you will have done at GCSE, so you will have done a very simple form of this, so this shouldn't be completely new to you. So the first thing you have to do is you have to find the piece of DNA with the insulin gene on it using some biological tools called restriction enzymes. And these then create little sections of DNA with what they call sticky ends. So in here we've got the enzymes, and what they do is they effectively cut the DNA for us, and that's all that we really need to know about at this stage. And from that they will be able to cut out, hopefully, the insulin gene. Now, in order to modify your bacteria, you're going to want to insert that piece of human DNA into a bacterial plasmid. So in order to do that, you're going to cut the bacterial plasmid with the same restriction enzyme, and that produces sticky ends which match the ones on the human DNA. Don't worry if all of this sounds a little bit unfamiliar to you, but if you just follow the overall context of it, and don't worry too much about the terms restriction enzymes and sticky ends, because that will become more familiar as we do the biotechnology and genetic engineering topics. So the bacterial plasmid then has been cut, and that's created these staggered cuts in the DNA, and we're going to insert the human DNA into the bacterial plasmid using DNA ligase, which is basically like DNA glue, and that creates a bacterial plasmid with the human gene inserted. So now we've got a plasmid with the human insulin gene in it. And that modified plasmid is called recombinant DNA, and all you need to do now is get that plasmid into the bacteria. Once you've got a bacteria that's picked up that plasmid, and bacteria like to pick up plasmids because plasmids usually carry antibiotic resistance, so there's a real benefit to the bacteria picking it up in the first place, so it's not difficult to get them to do it. Once they've picked up that plasmid, they will then follow the instructions on the plasmid and start to produce the human insulin as they express the gene. And if you grow that bacteria in a fermenter with large numbers of other bacteria and all the right things that you need to grow bacteria, the bacteria will start to secrete the insulin into the system, and you can harvest that and purify it, and then you can use it for people who are diabetic. So genetically modified bacteria has a lot of advantages over pig insulin. It's cheaper. Human insulin is less likely to cause allergic reactions. There are no ethical concerns about the fact that we're killing animals to harvest their insulin. And you can produce it in much higher quantities. It's not seasonal, it's not affected by other diseases. The potential for picking up diseases from other animals is always there, so all of those things are actually reduced. Some people could have an alternative treatment and maybe have a pancreas transplant, but there aren't very many transplants of this nature, and it's always the problem to get organs for transplant, and the risks of having to have immunosuppressant drugs when you have transplants are also a bit of a downside to it as well. And you've got the problems with rejection, which immunosuppressant drugs are trying to resolve. So that means you're always susceptible to other diseases. Other treatments. Sometimes some doctors have been trying to inject patients with pancreatic beta cells, and about 8% of the cell transplants are successful. So effectively what we're trying to do is we're trying to repopulate the pancreas with the cells which are no longer working. Stem cell therapy is thought to be a real potential for the future, and lots of work is going into that at the moment. Because stem cells are totipotent, they will have the ability to divide and specialise into any type of cell, and that includes beta cells. And what they've been trying to do is they've been trying to find out how you can turn a totipotent stem cell and encourage it to become a beta cell. So how do the cells know which type of cell to become? So they're trying to work on that process. And also they're also working on where the best location is to do this. Is it better to try and cause this specialization in a test tube, or can you get them to do it if you put them into the pancreatic tissue itself, for example? This just takes us on to a little bit about stem cell ethics. The way that you get totipotent stem cells is by extracting them from embryos, because it's only embryos that still have totipotent stem cells. As soon as embryos become more than a few days or weeks old, they stop being totipotent and start to become pluripotent. So you have to actually extract these very, very early cells. That means, of course, destroying the embryo, and a lot of people would be very concerned about the ethics of that. On the other hand, a lot of embryos are not required from fertility treatment, so people who have IVF, for example, usually produce more embryos than they need, and so quite a lot of them are destroyed anyway, because there's a limit to how long you're allowed to keep them. So you could argue that rather than having them destroyed, that you could use those totipotent stem cells to do something more useful. One of the benefits from that would be that the stem cell lines of a very small number of embryos could be used to treat a lot of patients, because you can grow those stem cells into large numbers. An alternative could be to extract some stem cells found from the umbilical cord. Quite often it's the umbilical cord blood that's useful. So, and that of course doesn't result in any damage to the baby. So looking at some of the advantages of using stem cells then. Firstly, donor availability. This is not such an issue. As long as you're prepared to use embryonic stem cells, then we can produce an unlimited number of beta cells from them. There's a possibility that you would get less rejection using these stem cells, but this isn't necessarily the full story. If stem cells are your stem cells, then your body is probably not going to reject them. However, if you use stem cells that belong to somebody else, so if you take an embryo, extract those stem cells, it's quite possible that those stem cells will trigger an immune response and the body will reject them and destroy them. One of the main benefits, of course, of stem cell advantage using stem cells is that people won't have to inject themselves with insulin, and their blood sugars will stabilise and they won't have all the health problems that are associated with diabetes. Some of the disadvantages of using stem cells: their availability and the ethical issues involved, which you've already covered. The potential to be rejected by non-self donors. Our ability to control the growth and differentiation of stem cells is still limited, and there is still a link then to the concern that this could lead to the formation of tumours, which is basically cancer, and so we need to be able to have a greater understanding in the way in which stem cell growth is controlled, otherwise it would lead to other health concerns such as cancer. So to finish off, we're going to have a look at a few questions. So have a look here at the questions on the sheet. What is type 1 diabetes? Describe how type 1 diabetes can be treated with insulin therapy. Give another way that type 1 diabetes can be treated and describe what this involves. Describe the different ways that type 2 diabetes can be treated. 3 advantages of using genetically modified bacteria, and describe how stem cells may be able to cure type 1 diabetes. Have a go at these questions first before you continue with this presentation for the answers. So what is type 1 diabetes? Autoimmune disease in which the body attacks and destroys the beta cells in the islets of Langerhans. Describe how type 1 diabetes can be treated with insulin therapy. For most people with type 1 diabetes, insulin therapy involves regular insulin injections throughout the day. Some people use an insulin pump, which is a machine which continuously delivers insulin into the body via a tube under the skin. And another way it could be treated is having islet cell transplantation, receiving healthy islet cells from a donor so the person's pancreas can produce some of the insulin, although they might still need some insulin therapy. Describe the different ways that type 2 diabetes can be treated. So mostly through lifestyle, a healthy eating, balanced diet, exercise, losing weight. And if you can't do that, then we go on to medications, which might include insulin injections as well as various other types of medications which control blood sugar. 3 advantages of using genetically modified bacteria to produce insulin rather than animal pancreases. Produces insulin uses GM bacteria is cheaper than extracting it from animals. You get larger quantities. It makes human insulin, which is more effective and less likely to trigger an immune response. And there's also the ethical region if people, for example, are vegetarian. Describe how stem cells may be able to cure type 1 diabetes. They could be grown into beta cells which could then be implanted into the pancreas with type 1, which means the person would be able to make insulin as usual. Here are a few more questions for you to have a go at. Again, as always, look at the questions, have a go at them before going on through the rest of the PowerPoint. Copy and complete the table to compare and contrast the differences between type 1 and type 2 diabetes. So we've got inability to produce insulin, no insulin produced or cells not responding properly to insulin. When does it develop? In childhood. Type 2 later in life. Period of development is fast in type 1, slow in type 2. Explain why people with type 1 have to constantly monitor their blood glucose concentration. So the answers must include any 3 of these. Blood glucose concentration will vary throughout the day as a result of diet and exercise, which of course is for everybody. Correct example given, so for example, increased exercise will cause blood sugar to go down, eating carbohydrates will cause it to go up. Plus any of the following. Knowing the exact blood glucose concentration ensures the correct dose of insulin is given. If a sufferer gives too low a dose, they will remain hypoglycemic, which can result in death. If a sufferer gives too high a dose, they can become hypoglycemic. This can result in a coma or unconsciousness, and any other sensible suggestion. Explain why some who suffer from diabetes mellitus who can produce insulin cannot control their blood glucose concentration. Any three from they suffer from type 2 diabetes. Their body cells do not respond to insulin or aren't sensitive to insulin because their cell surface receptors aren't binding to insulin. Cells don't take up enough glucose from the blood, also blood glucose concentration remains high. Discuss the advantages and disadvantages of different treatments for diabetes for 6 marks. 1 mark awarded for each correct stated advantage or disadvantage, but you have to have 1 advantage and disadvantage for each treatment. So here are some of the things. Insulin, advantages, readily available, and dose can be easily altered, it's cheap. Disadvantages, reliant on drugs, side effects, person has to inject themselves. Transplant, permanent cure, but no reliance on drugs. Disadvantages, can be rejected by a person's immune system. You have to have immunosuppressants, not enough donor organs available. Stem cells, permanent cure, don't need to take drugs, no risk of rejection, stem cells are readily available. Disadvantages, technology isn't advanced enough yet, and you have to obviously kill embryos in order to do that. And this brings us to the end of this particular presentation on diabetes, and also this ends the course on hormonal control. Thank you everybody for listening, and by all means ask any questions if you're finding any of it difficult. Thank you very much. There are a couple of videos which I will give you the links for. They're actually linked on here, but you won't be able to get hold of them from this presentation, so I will put those in the work for you so you can have a look at them.